Hello, folks. So what we're going to do today is make a big old drawing of everything involved in photosynthesis. So let me try to dim the lights just a little bit here to get rid of some of the glare that might make it more difficult for you to see. So hopefully this works with helping you see everything with our drawing. All right, so I'm going to start with black. What we need to do is make a huge box to represent like a plant cell, okay? So again, this will take almost all the space on a piece of paper. So you can use your own paper at home here, but you want to make some space on it, okay? Now, within that, we're going to make a huge chloroplast. This is way bigger than what you would typically draw in a chloroplast, but we need space. So this is a monster chloroplast that will take up almost all the space inside this plant cell, okay? So let's label what we've got here. Here, this is a plant cell, or we could say other photoautotroph. Photo means light, auto means self, and troph means feeding. So it's an organism that makes its own food. So we've got in this case, we'll just go with a generic plant cell. We're going to make a huge chloroplast inside of that. All right. Now, what are we going to add to it? Well, I'm going to move with green here, and we're going to make a membrane. This is our thylakoid membrane. So let's see. Again, in the thylakoid, this is going to end up making up most of the space inside your chloroplast. So we're going to make 5 little gaps here. 1, 2, 3, 4, and 1 more. And then we'll make a stack of membranes. It's called a granum. We'll make another little gap down here. All right. So here is our thylakoid membrane in green. Again, it looks like a stack, and we've got some gaps in here. All right. I'm going to add some green circles in a couple of spots here. And then we're going to add some blue circles. All right. So this is tough to see. Let me see if little around with the lights a little bit more helps with this. Okay. All right. So we've got the larger plant cell. We have the chloroplast. Within the chloroplast, we have these thylakoid membranes with green, and then I got some blue stuff going on here. So let's label these parts. This membrane is the thylakoid membrane. And this fluid inside of here fills up what's called the thylakoid space. Okay? So the thylakoid membrane creates this structure within the organelle, and then inside of it is the thylakoid space. Let's see. The green structures that we have here, I'm going to fill those in with some dots. And these green structures represent the photosystems. Now, we have photosystem 2, PS2, Roman numeral 2. And then there's photosystem 1, we use PS, Roman numeral 1. Don't pay attention to the number in terms of sequence. Again, it's the order in which they were found. Okay. Now, these first 2 blue structures that we see here, these are going to be electron transport chains. So they shuttle electrons along. Okay. And let's look at what these green dots represent. These green dots represent, I'll just put it out here because I've got space, chlorophyll. So chlorophyll is the green pigment that fills up the photosystems, and it what makes the chloroplast green, and it is what makes green plants appear green. Okay, so the greenness comes from these chlorophyll pigments that are embedded in photosystems, and the photosystems are embedded in the thylakoid membranes, and the thylakoid membranes are in the chloroplast. Okay, so that sort of gives you some context there. Now, what happens is we got the sun. Let's draw a little sun out here. I'm doing this in red. You could choose your own warm color. So rays from the sun strike these chlorophyll pigments. So energy from the sun, photons of light from the sun, strike these pigments, and the chlorophyll pigments absorb many wavelengths of light. When they do that, it excites electrons. So I'm going to draw in some electrons here, and do E- for my electron. And what happens is the electrons get bounced around between the chlorophylls until they reach a special pair of electrons that pass those electrons along to the electron transport chain. So electrons end up getting passed down the electron transport chain. Now, another photon of light strikes chlorophylls in photosystem 1, and then the electrons in photosystem 1 also get excited. And then those electrons get passed down the electron transport chain again. Now, what ends up accepting all these electrons is an enzyme embedded in the thylakoid membrane. It is called NADP plus reductase. Okay, reduction, oil rig. Reduction is a gain in electrons. So this NADP plus reductase takes the electrons and then reduces NADP plus and this proton to, draw this in black. And I'll put this in red to represent energy. NADPH. All right. So the photons of light, the energy from the sun, strikes these pigments in photosystems 2 and 1. It excites electrons that get passed along through NADP plus reductase, this enzyme, ultimately to NADPH. Okay? So we've basically taken some electrons and protons, put them in this molecule, an electron carrier. Now, an issue we've got, excuse me, is that these photosystems have lost their electrons, right? If the chlorophyll pigments have their electrons excited and they get passed along, eventually the electrons get lost. So you're going to have to replace them. So where are you going to get electrons? From water. It's something that's handy, available. So let's put water in here. I'm doing this in blue, so H2O. And what will happen is when water gets split apart, I'm going to draw my arrows in black here. The electrons, which I'll draw in red, get handed off to chlorophyll. So when you split apart water, the electrons are passed on to chlorophyll. The protons from water, when those get split apart, begin to concentrate in the thylakoid space. So now, as we split a bunch of water molecules apart, we begin to create a gradient of protons. And then the electrons are passed along, the protons get passed along. The oxygen from water, and I'll do this in blue, the oxygen is released as a waste gas. So collectively, all these events associated with light energy, they're called the light reactions. So in the light reactions, light strikes chlorophyll in the photosystems that excites electrons that get ultimately passed on to help create NADPH. To replace the electrons lost from the photosystems, water gets split apart. And when you split apart water, the electrons go to chlorophyll, The protons accumulate in the thylakoid space and create a gradient, and then oxygen is released as a waste gas. Now, in blue here, I'm going to draw an important enzyme, one we're familiar with. It's ATP synthase. Just write it down here. So ATP synthase, when it spins, we're familiar, it can take ADP, adenosine diphosphate, and an inorganic phosphate group, and it can make ATP. All right. So at the conclusion of the light reactions, what do we wind up with? We wind up with light energy converted into the chemical energy of ATP and NADPH. We also end up with water being consumed and oxygen being produced as a waste gas. All right. So at this point, we've converted light energy into chemical energy, but we haven't made any sugar yet. So that's where the Calvin cycle or the dark reactions, or sometimes they're called the light independent reactions, that's when the Calvin cycle takes place. What we do in the Calvin cycle is basically use this energy created during or produced during the light reactions, and we use that to make sugars. All right. All right. So here in black, I'm going to draw another cycle. Again, this is the Calvin cycle. And just like any other cycle, we start with an enzyme fixing carbon to a molecule. So our existing molecule is ribulose biphosphate. You don't need to memorize this at all. But there is an enzyme, I'll mark this in blue, called Rubisco. Sound like a cracker. Okay, Rubisco is an enzyme. And what happens is carbon dioxide, I'm doing this in black, carbon dioxide from the atmosphere joins in with the help of this Rubisco enzyme to RUBP, and it forms molecules that you don't need to know. What we'll do is just call them intermediates. So you form all these different intermediate molecules. And along the way, what we do is use ATP and NADPH to rearrange these molecules into these carbon-based molecules called G3P. And these are, I'm going to do this in black, 3 carbon molecules. So you take carbon dioxide out of the air, use an enzyme to attach that carbon into an existing molecule, use ATP and NADPH to rearrange those carbons into these 3 carbon sugars. And then those 3 carbon sugars will be rearranged into glucose. That 6 carbon sugar used in cellular respiration. Now, to regenerate the cycle, we are going to use a little bit more ATP to rearrange the molecules that are left back into RUBP. So ATP is used, NADPH is used. This will recycle ADP and NADP plus. And then these molecules can go back and be regenerated back in the light reactions. So as long as you have light, you can power the creation of ATP and NADPH. As long as you have ATP and NADPH and carbon, you can fix that carbon into these intermediates and use the energy you captured in the light reactions to crank out these sugars. Glucose then can go through glycolysis, and then the pyruvate can enter the mitochondrion, and you can use that to generate a bunch of ATP. Okay? So the whole purpose of photosynthesis is basically to convert light energy into the chemical energy of sugars. And then we can use the chemical energy in sugars to power the production of ATP. And then basically with ATP, you got a bunch of molecules that have this phosphate group that can hand off to other substances. When you hand off that phosphate group, it changes the shape of the molecule, and then it does work for the body. All right? Life. How spectacular, right? So that takes care of that. As you work through this, if you have any questions, please let me know. But we've got this marvelous process of taking light energy and turning it into food, using that to create food. All right, thank you so much. I'll awkwardly walk over here and turn things off. Have a great day. Good luck with everything.